So let's start with central dogma. We know that central dogma is about transcription translation, so let's delve into it. So what does unwind the twisted double helix DNA bit to form a section of separated DNA called the transcription bubble, so that what enzyme can fit in the DNA and transcribe it. So let's go from the end. We can see here that there's an enzyme that can fit in the DNA. Let's zoom in on this. So there's the enzyme that can fit in the DNA and transcribe it. And there's another enzyme that unwinds the twisted double helix DNA. So what unwinds the double helix DNA is the helicase. And this forms a transcription bubble, and you can see here that it is rewinding here and here. So as soon as helicase is finished unwinding, and RNA polymerase can fit in this transcription bubble and transcribe it. As soon as it's finished, then the DNA is rewound back into the double helix form, twisted double helix form. And yeah, that's the concept. So we should know that this section is called the transcription bubble. Helicase unwinds it, so makes the bubble, and RNA polymerase goes in the middle. We should keep in mind that it is the RNA polymerase that goes in the transcription bubble. So we can write it down on the side of our paper note or whatever you have in front of you, that RNA polymerase, RNA pol is for DNA transcription. So for transcription, we have the RNA polymerase going into the transcription bubble. Okay, let's go to this one. So we know that there is no thymine in RNA. It's uracil instead of thymine. So we should keep that in mind. And let's continue with, where are we? I'm just going through my desktop to find the note from yesterday. Okay, perfect. Now the base thymine is replaced with uracil in RNA, so that totally makes sense, and we should know that by heart. So if you don't know, just keep in mind that uracil is replaced with U is replaced with T in RNA. So sorry, I meant the opposite. T is replaced with U in RNA. So what strand is the double-stranded DNA strand used for transcription? It is complementary to mRNA. So the template strand is the strand that is in the double-stranded DNA used for transcription, and it is complementary to mRNA. There is another strand opposite to template strand, complementary to template strand, and it should be non-template or something else. We can see it. We'll definitely see it in the next few flashcards. So the template strand is the double-stranded DNA strand used for transcription, and it is complementary to mRNA. This is also important. So when we say that it is complementary to the mRNA, first things first, mRNA is the complementary strand that comes in front of the template strand. You make it, you transcribe it, and then transcription is over and the double helix structure goes back to the twisted form. So the unwinding is finished, the transcription bubble goes back to the twisted form of the DNA. And that mRNA is complementary to the template strand. We should also know about the reading directions and everything, so we'll see it here. There's another strand, which is the coding strand, and that is the DNA that is not used during transcription. Okay, perfect. So the non-template strand is called the coding strand, but it is identical to mRNA. Right, makes sense. So, but you just in RNA you just substitute T with U, right? Perfect. So we have the template strand and the coding strand. Probably we can also keep in mind that the coding strand is similar to the codons. We'll see that very soon. The star codon is AUG, and it codes for methionine. Remember this because usually school starts in August. So AUG, August, it starts, and makes sense, and it codes for methionine, however you call it. The ribosome begins translation by matching codons, 3 nucleotide long sequences, with anticodons on tRNA, right? Let's see. So the So the ribosome, also make sure that you turn off your phones to avoid any distractions. That's what I just had to do. The ribosome begins translation by matching codons. Okay, so now we're talking about translation here. And the ribosome is the thing, the protein, that is the structure, the organelle that does the translation. Remember, ribosome was making proteins, synthesizing proteins, and then it was transferring it, relaying it to the Golgi apparatus for the packaging and modifications that's needed, and finally exocytosing it to anywhere that you need to send the protein, right? But before that is the ribosome that is also in the rough endoplasmic reticulum that begins translating by matching the codons with anticodons on tRNA. So these tRNA molecules are carrying amino acids, and as they get recruited, they drop off their amino acids to be added to the growing polypeptide chain. And we can see animations on this later on, but I have studied this before, so I know more or less what this is talking about. The key point here is that to know that the ribosome begins translation by matching the codons, the 3 nucleotide sequence, with anticodons on tRNA. So the anticodons are on tRNA, and the codons are on the mRNA, where ribosome is the structure where this whole translation takes place in. So it is in the ribosome where this codon on the mRNA is matched with anticodons on tRNA, and it is the ribosome that does this, right? The pairing between the third nucleotide of a codon and anticodon experiences wobble. So wobble is what causes the third nucleotide on the codon to mispair with the anticodon, right? This is one of the reasons why multiple codons can code for the same amino acid. So one of the reasons that multiple codons can code for the same amino acid is to avoid any mutations that could possibly happen with the wobble, right? The mature mRNA leaves the nucleus and is turned into a protein by a ribosome in a process called translation. So the mature mRNA, so this implies that mRNA is maturized in the nucleus, and it leaves the nucleus and turn into a protein by a ribosome in the process called translation. The fragments of DNA that are created on the lagging strand are called Okazaki fragments. So, what is the lagging strand? Now, we should know that there's also lagging strand here. Are we talking about translation or transcription here when we're talking about Okazaki fragments? We will figure that out soon. So let's move forward, but we just write here is Oka I hope you are saying this well. Yes. So is Okazaki fragments I know this is not grammatically correct, but is Okazaki fragments happening in transcription or translation? Probably our question is going to be answered in the following flashcards. The first step of replication is to break the hydrogen bonds between base pairs in DNA by an enzyme called helicase. So it's good to know that the very first step of replication, as we know it as transcription, is to break the hydrogen bonds between base pairs in DNA. So we need to know where these hydrogen bonds are in base pairs and what is the whole structure of the DNA, including its base pairs, that it is broken down through the hydrogen bonds by an enzyme called helicase. So instead of the resulting section of separated DNA strands being called a transcription bubble, it is now called a replication fork. Okay, so transcription bubble is when it is happening during transcription, right? But so we are now talking about replication. And replication is different than transcription. So in transcription, you just want to make an mRNA. So you just make it from the template strand, if you remember. And it is done. You don't need to transcribe or make anything from the coding strand, which is the opposite strand of the template strand. But here in replication, it is a different scenario. You want to make copies of both of the strands of the DNA, right? So first things first is that in replication, this is basically about, we're talking about, I'm pretty sure, mitosis and meiosis, right? And it is important to know that in replication, the first step is to break the hydrogen bonds between the base pairs in the DNA. So, and this is done by helicase, right? But this is not called so whatever forms like the transcription bubble, it's not called the transcription bubble. That's for transcription. In replication, we call it the replication fork. The enzyme that reads and transcribes DNA is called RNA polymerase. So we know that in transcription, it is the RNA polymerase that is transcribing the DNA, the template strand, right? So we need to know the stop codons. There is a mnemonic here, but I have my own way of memorizing this. I like to memorize the stop codons with yoga, which is UGA. It starts with UGA, and then you swap the position between G and A, so it's UAG. So we have yoga, and then you swap it. So I go by reverse, right? The opposite direction, yoga, and then you swap these two. We have UAG, and then you have UAA. You just kind of make it wobble on the third nucleotide, and I just like to memorize it like this. But you can memorize you are annoying, you are gross, and you go away. Okay, that's a good mnemonic. Not gonna lie, that's a good mnemonic. But I'll need to just think about. I was thinking about something. A question about the hydrogen bonding between the base pairs. So, how does helicase break the hydrogen bonds in helicase? Keep in mind that I'm not answering these questions right when I write them, because I want to first have a overall glimpse, and there is a second reason that it enables me to have do a space repetition on these concepts. So I'm bringing them up again, which if you remember from the memory chapter, it is a rehearsal practice. So I'm able to bring back the concept, and that enables me to rehearse it and retrieve it from my long-term memory to my short-term memory. And the more you do it, the faster you can bring it up, and better for the exam. Faster. Termination occurs when the ribosome reads a stop codon. Yes, we know that. So that is what the stage is called, termination, right? Some viruses called retroviruses use an enzyme called reverse transcriptase to make DNA from RNA. So this is very interesting. When you have a DNA, when you go, this is the whole central dogma, right? So you transcribe the DNA into RNA and you translate the RNA into protein. Now, funny here that viruses, a specific one, which is called the retrovirus, retro means that it goes in the reverse direction, right? Whatever is retro, we have also retrospective studies that we need to know for the MCAT. It means that it is kind of reverse chronological. It is reverse chronological, and this reverse chronology, it's hard to pronounce it, right? Reverse chronological, and this reverse chronology is reverse chronology is to make DNA from RNA, right? And this enzyme is called the reverse transcriptase. So they have an enzyme which is reverse transcriptase and is able to do this. RNA polymerase, which, if you remember, it was in the transcription bubble. It is RNA polymerase that is putting the base pairs in front of the template strand and making up the new strand for transcription. This is like probably talking about the function, right? It binds to a region on the DNA called the promoter region. So RNA polymerase starts in the transcription, it starts putting in the base pairs with attaching to the promoter region. And this promoter region is often a TATA box. So, and TATA is basically the fact that it's TA sequences, right? That sequence is the promoter region. Promoter TATA. RNA is single-stranded most of the time, whereas DNA is double-stranded. So, of course, RNA is one-stranded. There's also uracil instead of thymine in RNA, and it is a ribonucleic acid, but DNA is deoxyribonucleic acid. So this is a ribose sugar and this is a deoxyribose sugar. Deoxy means that it has one less oxygen. You can see here that the ribose sugar has OH here, and deoxyribose has only hydrogen here, and it is on the second carbon, right? So you start from here, 1, 2, 3, 4, 5, and it's a pentose. You can make sure to ask, is ribose or deoxyribose a pentose sugar? RNA is single-stranded. Yes, we read that. RNA has a hydroxyl group on the second carbon. The DNA doesn't, hence the deoxy prefix of DNA. See? So it's on the second carbon, as I told ya. On the lagging strand, DNA polymerase can't read the strand from 5' to 3'. So we are talking here about probably replication, which is because it's lagging strand, DNA polymerase, and reading from 5' to 3'. And the direction the helicase is unzipping the... Oh, so we have I believe in replication is called the sense strand. So instead, a primase adds multiple primers so the DNA polymerase can latch onto those primers, complete a small section, then move upstream to the next primer and complete the next small section, and so on and so forth. These fragments of DNA are called Okazaki fragments. Cool. So we know that the lagging strand is the 5' to 3' strand. The 3' to 5' strand is the sense strand, I believe. I need to be double checked, but it is in the lagging strand because the helicase is unwinding DNA, and in the lagging strand is the 5' to 3' direction, and the DNA polymerase cannot read from 5' to 3'. Therefore, it is the Okazaki phenomena that happens. It is Okazaki fragments. So DNA polymerase goes on the primers that primase, another enzyme, has added to the as complementary to the lagging strand, and DNA polymerase starts from the first primer and then moves upstream. By upstream, I believe we are referring to the direction right. So it moves to the right and then completes the next small section, the next Okazaki fragment, and so on and so forth until it is finished. On one of the strands, DNA polymerase can freely slide from 3' to 5', adding nucleotides as helicase unzips the parent strand. So it is the parent strand, the main one for replication. Because of this smooth process, this is called the leading strand. So we have, forget about sense probably, is not a thing. So we have leading strand and lagging strand. We can ask here, is leading strand called the sense strand as well in replication? How about the lagging strand called the nonsense? I know we have nonsense mutation, which results in the nucleotides coding for the stop codon instead of coding for another protein. So we should keep that in mind. mRNA is transcribed in the nucleus. So we're going back and forth between transcription and replication. So for transcription, mRNA is transcribed in where? The nucleus. That's right. Is translation taking place in the nucleus too? What is translation? It is the RNA, the mRNA translating for protein. Where do we make proteins? It is the ribosome. Is ribosome inside the nucleus or outside the nucleus or on the nucleus. We know it is in the rough endoplasmic reticulum, and rough endoplasmic reticulum attaches to the nucleus, so it should be outside of the nucleus, but we can still ask it. Is transcription happening inside the nucleus? We know yes, nucleus. And is translation happening outside? Guanine is complementary to cytosine, yeah, GC. There's also the GC cap, something like that, for the exons and introns. You'll face it soon. For now, during elongation, during which stage tRNA molecules are carrying amino acids, and as they get recruited, they drop off their amino acids to be added to the growing polypeptide chain. This occurs in the ribosome. Yes, we know that. It is during. So what is elongation? It is basically the amino acid sequence elongating, right? So the tRNA molecules are carrying amino acids, and as they get recruited, they drop off their amino acids to be added to the growing polypeptide chain. We're talking about the primary structure of the protein here. Literally how the proteins are made. DNA molecule with a high concentration of CG nucleotides is going to be stronger than one with a higher concentration of AT nucleotides. That is important to know because CG has, I believe, a triple bond, but yeah, the number of hydrogen atoms. So A and T have 2 hydrogen bonds between them, while C and G have 3 hydrogen bonds. And the number of hydrogen bonds between those molecules is what determines the strength and stability of the DNA molecule, for sure. So we know that DNA is basically the leading strand and the lagging strand attaching together, the parent strand and the daughter strand attaching together, right? The connection between these base pairs is with hydrogen bonds, and C and G have 3 hydrogen bonds between them, which is stronger and more stable than A and T, which have 2 hydrogen bonds. DNA is transcribed into RNA, which is translated into proteins. This is the essence of central dogma. An enzyme called primase attaches to one of the strands of DNA and reads a small section of it, all the while attaching complementary RNA nucleotides to that small section. So what does primase do? It attaches primers. It attaches to one of the strands of DNA and reads a small section of it, all the while attaching complementary RNA nucleotides to that small section. So it attaches RNA nucleotides. This is also important to know. This is called the primer, where it is used on the lagging strand, and thereafter the Okazaki fragments happen. An enzyme called exonuclease eventually comes and removes all the primers, while DNA polymerase makes another round, filling in all the gaps that used to be primers. So we have an enzyme exonuclease, which comes and removes the primers, while DNA polymerase makes another round, filling in all the gaps that used to be primers. So DNA polymerase not only goes through the transcription bubble to do the replication, to put in the base pairs in front of the leading strand, and also doing it for the Okazaki fragments, but it also comes back at the end to replace the primers and make the correct sequence, the correct base pairs for to replace primers. An enzyme called binds to the primer, then slides down the DNA strand from the 3' to 5' direction, adding DNA nucleotides to the template that the original DNA strand provided. It is the DNA polymerase that binds to the primer and then slides down the DNA strand from 3'. So does DNA polymerase add nucleotides? So does DNA polymerase add nucleotides in upstream or downstream of either template leading or non-template lagging. Strands. Okay. But we need to know here for now that DNA polymerase binds the primer. It then goes downstream, down the DNA, down the DNA strand from the 3' to 5' direction. We're talking about the template strand here, adding DNA nucleotides to the template that the original DNA strand provided. Is template strand the same as the parent strand? Adenine A is complementary to thymine. We know that. A signal sequence, sometimes called a localization sequence, is a marking that tells the protein to go to a specific organelle. So what does tell the protein to go to a specific organelle? It is the signaling sequence, or sometimes called the localization sequence. The AAMC has used the term nuclear localization sequence on exams frequently. This just describes one of those signal sequences that is telling the protein to go to the nucleus. So if there's a name of the organelle before the localization sequence or the signal sequence, it means that the protein is destined to go to that location, to that organelle. So the nuclear localization sequence means that the protein is destined to go to the nucleus. A single codon codes for one amino acid. Yes, it doesn't code for more than one amino acid. But one amino acid is coded by multiple different codons. It can be coded by multiple codons. A single amino acid can be coded for by more than one or multiple codons. A nucleotide consists of, we know that it has the base pair, it has sugar and it has a phosphate. So not base pair. Forget about that. Base pair is the basis pairing. So it is like, for example, C pairing with G. But a nucleotide by itself, which is the nucleotides are the bases that are paired, right? So the nucleotide must have an amine group, which is the nitrogenous base, right? It has the base adenine. It has an amine group, the nitrogenous base, and it has a sugar and a phosphate. So the phosphate is PO4 2 minus or 3 minus, and there's the sugar. What is the chemical formula of phosphate? And is it PO4 2 minus or 3 minus in the nucleotide? Four types of nucleotides that can be in DNA: adenine, guanine, thymine, and cytosine. Boom, we're finished with I'm just glancing through the previous chapters. Oh, this one. Should we go over classic conditioning for a second? Quickly. We have unconditioned response, which is the response to the unconditioned stimulus. So what could be an example? For example, the dog is not conditioned to the bell ringing when the food is presented. Am I right? It is natural, like dogs salivating to food. The unconditioned response would be salivation to the food. Oh, so this is not conditioned. The conditioning would be with the conditioned response. It would be the same as unconditioned response. So in case of the dogs, it is the salivation. The only difference is what elicits the response. So we should know that the response is salivation in both scenarios, but what elicits in the conditioned response is the bell ringing, but the unconditioned stimulus for unconditioned response is the food being presented. We have spontaneous recovery. So if after extinction, we should know what extinction is. It's right here. Extinction is a cessation of association once one has already been made. So it stopped associating, or basically the conditioning. Usually an extinction occurs if the conditioned stimulus is presented too many times without pairing it with the unconditioned stimulus. So the conditioned stimulus being the bell. If you ring the bell too much and you don't provide the food anymore, the dog stops to salivate. So the behavior is extinct now. So if you continuously presented the sound without also presenting food, eventually the dogs would stop salivating to the sound. So spontaneous recovery is that if after extinction the unconditioned and conditioned stimuli pair together, there is a chance for the association to be instantly reacquired. So if you suddenly pair the bell ringing with the food, then there's a chance that the association is instantly reacquired. So the dog immediately starts salivating, and this is called a spontaneous recovery. We have generalization. So sometimes an association can be generalized to similar stimuli as the conditioned stimuli. And it's like ringing any bell or doorbell or the church ringing. And then the dog starts salivating. It's basically generalization. So if the bell ringing was a sound that the dog did the association with, perhaps the sound of a wind chime would also make the dog salivate. So typically, for generalization to occur, the stimuli have to be pretty similar, like the sound of the bell ringing and the wind chimes. We also have discrimination. It is deciphering between similar stimuli to determine whether we have an association to that stimuli or not. So if the dogs are able to distinguish between a bell ringing, which they would salivate to, and wind chimes, that is, which they would not, that is called discrimination. So they have successfully discriminated the stimuli. It's kind of the false error and accurate error. I don't know what I'm talking about. I think you know what I mean. We had the false positive, the real positive, the false negative, real negative, something like that. So if you're able to discriminate, you're telling the difference between the unconditioned and conditioned stimulus. Conditioned stimulus is the stimulus that does not naturally elicit the desired response. So it is the bell ringing. The conditioned response is the same as unconditioned response. The classical conditioning versus the classical conditioning is basically Pavlov experiment. This is associating a naturally occurring response with a stimulus that did not initially elicit that response. So Pavlov's dog. And so yeah, classical conditioning. Let me just say it by myself. It's when you when you are attaching a conditioned stimulus with an unconditioned with a response. So a naturally occurring response, which is salivation, with a stimulus that did not initially elicit that response. We have acquisition, which is the process of having learned an association. Think of it as acquiring an association. I have to jump into a meeting and I'll see you soon. Let me just finish here. We have the unconditioned stimulus, which is something that elicits a natural response out of the subject. So Pavlov noticed that dogs salivated whenever food was near. This is a natural response by the mouth to secrete more digestive enzymes in preparation for food. The unconditioned stimulus would be the food. So yeah, acquisition is basically just learning that behavior. Learning the association, basically. It's called acquisition. So if the dog is associating the bell ringing with the bringing of food, then that is when acquisition is taking place. So you are acquiring it. You're learning that. And unconditioned stimulus is something that elicits a natural response out of the subject. So, yeah. Perfect. We'll go to the enzyme kinematics soon. So for now, peace.